Hi everyone, today we're going to be talking about the mitosis cell cycle. The mitosis cell cycle is a form of asexual cell reproduction where the cell divides into two identical daughter cells, which start with the exact same process over again. It is needed for growth, repairing injuries, and replacing old cells. There are 4 checkpoints: G1, S, G2, and M. G1, aka the first gap phase, is where the cell grows by copying the organelles and proteins for the later steps. S is where there's a copy of DNA synthesized in the nucleus by replicating chromosomes to become double-stranded chromatids. It also duplicates the centrosomes. G2 is the second phase gap, and there is more cell growth, synthesis of proteins and organelles, and the organization that connects for preparation of mitosis. M, the next step, is where the M checkpoint consists of the stages PMAT that lead to cell division. G0, the resting phase, is where the cell does not divide, nor does it prepare itself to divide. Now the first phase for the mitosis cell cycle is interphase. This is the longest stage of the cell cycle where the cell prepares itself for division. It includes the checkpoints G1, S, and G2. The next phase is prophase, where the chromatin begins to condense into chromosomes and centrioles form the meiotic spindle, made of microtubules, between the centrosomes as they move apart. The nucleolus breaks down. In late prophase, prometaphase, the nuclear envelope breaks down. The meiotic spindle grows further and begins to organize the chromosomes. Metaphase is where the spindle captures chromosomes by attaching to the centromeres and lines it up in the middle of the cell.
In anaphase, the shortening of the spindle fibers separate the sister chromatids to the opposite poles, ensuring that each daughter cell has a complete set of chromosomes. The cell also begins to elongate and pinch inwards at the midline. In telophase, the mitotic spindle is broken down, 2 new nuclei are formed, and the nuclear membrane and nucleoli reappear. The chromosomes decondense back into chromatin. Finally, in cytokinesis, the division of the cytoplasm forms 2 new identical daughter cells, completing mitosis. How does mitosis connect to cell signaling? Well, regulatory proteins called cyclins control the timing of each stage of the cell cycle. These ligands bind to enzymes called cyclin-dependent kinases, the CDKs, and create the cyclin-CDK complex. The activated enzyme adds negatively charged phosphate groups to other molecules through phosphorylation, triggering other proteins and signaling the cell that it is ready to pass into the next stage. Once the cyclin degrades, it deactivates the CDK and signals an exit from the current stage. Thank you for watching.